there's more to protein structure than just the simple polypeptide bonds, and we'll look into that because things like temperature and low pH for acids or a very high pH for bases can affect proteins and denature them so they don't have the same properties they have anymore. Now, this is ideal in certain situations, and we'll talk about those a little bit, but most of the time when you're talking about bodily functions, it's not such a good idea. So here's an example of one of our polypeptide chains. So just the order that these amino acids are in is called the primary structure. That's the order these get put in. Now what happens is, because some of these are have different types of charges on the outside edge, you might get, and we know amino acids have nitrogen with some hydrogens on it, you might get hydrogen bonds pulling certain sections of these chains together, much like the two helix in a double helix of DNA are pulled together. That can cause what we call to be a secondary structure. Even furthermore, you start to get a 3D tertiary structure, and then the complex of protein molecules all together in a couple different proteins and how they join together is what is called your quaternary structure. So we've got primary, secondary, tertiary, and quaternary structure, and they're based off of these linkages between different molecules, mostly because of hydrogen bonds, sometimes just because of charges from the various bonds in the outside as well. Well, what happens is this complex structure we have here, which is for hemoglobin, if things get messed up one bit and this gets even just bumped over to the side just a little bit, it won't carry on the same function that hemoglobin does, and it won't hold oxygen as well. Hemoglobin is very important in our blood. It's what holds oxygen. And so if it gets bumped a little bit and this shape changes, it may not hold oxygen anymore. And that's one of the things that happens in sickle cell anemia, is the hemoglobin gets distorted and it can't hold oxygen anymore. Now one thing is, by heating things up with temperature, you can see that this quaternary structure here starts to unravel. And once again, the protein can't do the job that it normally does. And remember, proteins basically are our codes for our traits. And so if the protein changes and doesn't function the same, then the traits won't be expressed in our body the same. One of the easiest examples for that is that of an egg. We know egg whites are typically very clear, and if they get heated up, then you've got a different color here, and the properties of that egg white change. It goes from jelly-like to stiff. It goes from clear to white, and it doesn't function the same way that it normally does. And one of the things when you think about this with the egg is there's no going back here. You can cook an egg, you can put it back in the fridge, but it's not going to turn back into regular egg whites like you had before, that clear substance. If you cook chicken meat, the muscle in the chicken denatures and goes from kind of that pinkish translucent to a much more tan or kind of off-white color and gets much more firm, and there's no changing it back. And so an example here is you can see like this would be the structure of some of your proteins, and then they get unraveled. So high temperature is one thing that can unravel these proteins. The other thing is very low pH, which is like an acid, and very high pH, like a base. And so if you wanted to, you can try this at home. You can get an egg, put it in a bowl, get the egg whites out. You can take the yolk out if you want, just so that you can prove to yourself that yes, it is the egg whites. And then dump a whole bunch of vinegar in there and mix it up for a while and see what happens. And I think what you'll see is some of it starts to denature, and you get some of this white stuff in there from the acid that affects the egg. So that's something you could try at home to show your own protein denaturation. I think for me, I'm going to go home and cook some chicken on the grill, and that's how I'm going to do my denaturation experiment. So hope you enjoyed learning a bit more about proteins and how if they get extreme temperatures or low or high pHs, that they cannot function like they normally do.